So yeah, as Carol said, I am a PhD candidate in Maren Friesen's lab. I am in the molecular plant sciences program, and I have been doing some research on Piola and really now how management impacts the microbiome of Piola. So for those who aren't familiar with Piola, Piola is intercrop pea and canola, and it is a more efficient cropping system. And so what that means is that trying to move this bar out of my way. Okay, there we go. So what that means is that there's a 63% increase in productivity compared to monoculture pea and canola. And it is cited as having one of the largest increases in productivity compared to other intercrops. So what that looks like in terms of monetary value is that Piola earns $95.82 more per hectare than pea monoculture, and it also earns 27 more dollars per hectare than canola monoculture. Part of the reason for this increase in profit, in addition to its increase in productivity, is it needs fewer synthetic inputs. So it can competitively exclude weeds, so there's less need for herbicides. It performs worse with fungicide applications in some cases. Some have seen a decrease of $69.35 per hectare with fungicide application. And in some cases, fertilizer application, specifically of nitrogen fertilizer, decreases its success. So there was a group that saw that Piola with 67 kilograms of nitrogen per hectare saw a decrease in $21 per hectare in profits. And will it let me move on? There it goes. Okay. So in a legume and non-legume intercropping system, there are functions that the microbiome performs that provide the plants with nutrients. And so that could be part of the reason that we see decreases in the productivity with nitrogen application and fungicide application. And these can happen through indirect and direct means. So these indirect means are seen, oops, now it's changing on me, in the red boxes with root exits from plants. They can sometimes release nitrogen from the soil, then it's able to be taken out by the plant. You can get the degradation of nodules, which, for those that don't know, are a structure on the root that has nitrogen fixing bacteria. And so when those degrade, it releases nitrogen into the soil, in addition to, even if the root tissues of the legume are degraded, that can also release extra nitrogen into the environment. The bacteria in the soil also are able to regulate the activity of soil enzymes, which can then increase the end mineralization. More of a direct sort of transfer of nutrients can happen through arbuscular mycorrhizal fungi, through like mycorrhizal networks, which can facilitate direct transfer of nutrients between plants, assuming both plants are mycorrhizal. So really understanding how the microbiome contributes to Piola's success with minimal inputs will help us develop better and more successful intercropping systems in the future. So then that leads us to our question that we addressed with this data, which is, does nitrogen fertilization have an impact on the Piola soil microbiome? We predicted that higher levels of nitrogen fertilizer application would cause a shift in the microbiome, specifically through decreasing nitrogen fixing taxa, which are those different kinds of bacteria. Since plants will be disincentivized to interact with them, since if the plants have all they need, they're not going to want to put energy and their own resources into forming this relationship with these nitrogen fixing bacteria. So really, before I jump into the data, I just want to define a few ecological terms that we use that maybe some of us aren't familiar with, that will be relevant. So first, I'm going to talk a little bit about community diversity. And so I have a graphic here from Oregon State University of different plants from a rangeland ecosystem. And what you see is that you have different kinds of plants represented And then you also have the number of those different kinds of plants. So those both are contributing to community diversity. And then in addition, like the different types of plants make up the community composition. We also will be talking a little bit about the core microbiome, and the core microbiome is made up of bacteria that are found across different samples. So in this case, I have three different kinds of plants, and then their different microbiomes were sampled, and bacteria that were found across all of those three different microbiomes are considered to be core in the core microbiome. Then we have differentially abundant taxa, which we'll be talking about. And those are basically bacteria that are found in higher numbers than in one sample versus another. So this blue bacteria is found to be more abundant in the pea sample than the canola sample. So to begin to address that question, we did collect soil samples, which were the top 10 centimeters of that bulk soil. And then we collected rhizosphere samples, which is a layer of soil in direct contact with the root. These samples were collected in the summer of 2021 at the WSU Research Farm near Davenport, Washington. With Piola, there were three different nitrogen application rates that were used. And then here I have noted the rate of fertilizer that was applied to the canola and pea monoculture. There was also a background fertilization rate of phosphorus of 33 kilograms per hectare. So starting to look at the soil microbial communities, we saw that there were no differences in the soil microbial communities when assessing that community diversity, the community composition, and the differentially abundant taxa. When we looked at the core microbiome of the bulk soil, we did see that Piola has unique core members across the different fertilizer treatments. So these bacteria were not found in the pea and canola monoculture core microbiomes, and then they also were not shared with some of the other Piola core microbiomes. So you look here at the Piola that was at 67 kilograms of nitrogen per hectare, there are 23 bacteria that were unique to its core microbiome. When we look at the 33 kilograms of nitrogen per hectare fertilization rate, there are 16 bacteria that are unique to that core microbiome. And then at 0 kilograms of nitrogen per hectare, there were no bacteria that were specifically unique to that group. However, that there were bacteria that were shared amongst specifically all three of the Piola core microbiomes across fertilizer treatment. I also will note that there were core bacteria that were shared with the pea and canola monoculture core microbiomes. So those bacteria were consistent across every single core microbiome. So when looking closer at some of those unique core bacteria in the soil, we found that they were important to some plant health and nutrient cycling aspects. So when we look at Piola at 33 kilograms of nitrogen per hectare, there was a unique Mucilaginibacter, which aids plants in contaminated and stressed environments and is a growth promoter. Piola at 67 kilograms of nitrogen per hectare had a unique Brady rhizobium, which members of that group contain nitrogen-fixing bacteria. There was also Amycoloptosis, which was unique across all of the three Piola core microbiomes. So again, that was not seen in the pea and canola monoculture core microbiomes. And it is known to produce antibiotics and aid in remediating heavy metal polluted soils. So then moving on to the rhizosphere, We saw that fertilizer treatment did not impact the community diversity, but the cropping system did. So what we see here is that the canola microbiomes, regardless of intercropping, were the most diverse microbiomes. Then intercropped pea comes in second, and then the pea monoculture rhizosphere microbiome was the least diverse. Now, it doesn't necessarily mean that it's better that the intercropped pea the intercropped pea microbiome is more diverse, but it does show that there was a shift, at least in that community. So similar to what we saw in the soil core microbiomes and the rhizosphere core microbiomes, we also saw that the Piola has unique core members across fertilizer treatments. So what we see here is that at the 67 kilograms of nitrogen per hectare rate, the intercropped pea core microbiome had 7 unique core members, which is the most that we see across all the Piola core microbiomes. This group also had the largest amount of bacteria in its core microbiome. Then when we look at intercropped canola at that same fertilization rate, there are 3 core bacteria that were unique to that core microbiome, and it had the second largest core microbiome. When we look at the 0 kilograms of nitrogen per hectare rate, we see that intercropped pea does not have any unique core members like we saw with bulk soil. And then at that same fertilization rate in the intercropped canola, we only see 1 unique core member. There is a bacteria that is unique to all the Piola core microbiomes, regardless of fertilizer treatment, and if that was the pea-associated rhizosphere, the canola-associated rhizosphere. And then again, there were core members that were shared with the pea monoculture and the canola monoculture core microbiomes. So there are, again, bacteria that are very consistently part of that core microbiome. So when we look again at these core bacteria that are unique to the Piola, specifically in the rhizosphere this time, we see that they are again important to plant health and nutrient cycling. So again, we see Mucilaginibacter being unique to the Piola microbiomes. And this aids, again, plants in contaminated and stress environments and is a growth promoter. We also saw a species of Rhodantibacter, which was found to be unique to intercropped pea core microbiomes. So it was not seen in the canola core microbiomes, the intercropped canola core microbiomes, or the monoculture pea and canola core microbiomes. And these bacteria are known to contain nitrate-reducing members, and they can survive in acidic and metal-contaminated environments. So now moving on to looking at differentially abundant taxa in the rhizosphere, we saw that between pea and canola in monoculture, there were 12 differentially abundant taxa. Interestingly, there was this group highlighted in the green box, which is a group of rhizobia that were more abundant in pea. Now, when we move on to look at intercropped pea and canola and comparing the intercropped canola rhizosphere to the intercropped pea rhizosphere, with no fertilization, with no nitrogen fertilization, we see that the rhizobia are no longer differentially abundant between the pea and the canola, which could suggest maybe there's some sort of movement of this group towards each other in those microbiomes. When we look at the intercropped pea and intercropped canola microbiomes with fertilizer application, we see that there were no longer any differentially abundant taxa between these two. This suggests again that there's a even larger movement of these rhizosphere microbiomes towards each other with intercropping and with these fertilizer these nitrogen fertilizer additions. So then to really sum that all up, the key takeaways from here are that intercrop P in the rhizosphere experienced an increase in diversity with intercropping. So that doesn't, again, it doesn't necessarily mean it's better for being more diverse, but it again it does show that there is a larger shift in that microbiome when it's grown with pea. I mean, when it's grown with canola. The core microbiome is impacted by fertilizer treatment, but contains unique members from monoculture that impact plant health and nutrient cycling. And then we see differentially abundant taxa are impacted by intercropping and fertilizer treatment in the rhizosphere. So again, that meant that there were no differentially abundant taxa with nitrogen treatment. And that group of rhizobia, they're no longer differentially abundant when we are intercropping